Hello, I am Dr. Anshul Garg with interesting and learning episode from one of the important unit of Biotechnology, which is prepared by Dr. Rupal Dubey, Assistant Professor, Guest Faculty, Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, Central University, Sagar, Madhya Pradesh, India. In this episode, we will continue our discussion about enzyme immobilization. In this episode, we are going to discuss other aspects related to enzyme immobilization such as need for plant cell immobilization, methods of plant cell immobilization, and also some enzymes in detail. This lecture is divided into 3 modules. Module 1 is about plant cell immobilization. Module 2, methods of plant cell immobilization. And Module 3 is about study of different enzymes. Let's start today's lecture with first module, which is about plant cell immobilization. Need for plant cell immobilization. Plant cells are characterized by large size, sensitivity to shear and oxygen, and need of cell-to-cell contact for metabolic production. The secondary metabolites are triggered by short periods of stress in cultures. Immobilization can overcome many of the limiting factors of suspension cultures with the distinct advantages of easier operation of biocatalyst from the product and also being amenable for biotransformation of low value compounds to high value products. Now coming on to different types of immobilization. First is direct intracellular binding due to natural affinity, that is adsorption, adhesion and agglutination. Second is covalent coupling on otherwise inert matrices. Third is intracellular connection via bi or polyfunctional reagent by cross-linking. Fourth is mixing with suitable materials, changing their consistency with temperature, that is embedding. And fifth is physical retention within the framework of diverse pore size and permeability, that is by entrapment or micro encapsulation. The choice of a suitable immobilization system is determined by the following requirements. The polymer material used for immobilization must be available in large quantities. It must be inert, non-toxic and cheap. It must be able to carry large quantities of biomass and its fixing potential must be high. The immobilization process must not diminish enzymatic activity of biological catalyst. Manipulation of the biological catalyst must be as simple as possible, as studied by Hall et al. In 1988. Now coming on to methods for plant cell immobilization. First is gel entrapment by polymerization. A monomer or a mixture of monomers is polymerized in the presence of a cell suspension which is entrapped inside the lattice of the polymer. The most common example is polyacrylamide. The method is based on the free radical polymerization of acrylamide in an aqueous solution. As the linear polymers are soluble in water, they have to be insolubilized with bifunctional compounds such as N,N-ethylene bisacrylamide. The free radical polymerization of acrylamide is conducted in an aqueous solution containing the cells and the cross-linking agent. Polymerization is commonly carried out in the absence of oxygen and at lower temperature such as at 100 degrees Celsius to avoid damage to the cell during the operation. An initiator N,N-tetramethylene diamine, that is TEMED, TEMED, is used. Both the initiation and the cross-linking agents are toxic to the cells and therefore their viability can be lost. For example, Staphylococcus, Roseus and Silybum marianum. Next method is gel entrapment by ionic network formation. In this method, polymerization of polyelectrolyte is achieved by addition of multivalent ions. The most common method is the entrapment in calcium alginate. This is a non-toxic process in which sodium alginate solution containing the cell suspension is dropped onto a mixture of counter ion solution such as calcium chloride. A uniform, spherical and highly microporous structure results which retains the cells. The method may be inconvenient over long run in media containing calcium chelating agents such as phosphates and certain cations such as magnesium ion that may accelerate disruption of gel by solubilizing bound calcium ion. Carrageenan is also a suitable matrix for the immobilization of plant cells. The gels of calcium and potassium carrageenates are very stable at room temperature and pH above 4.5. Next method is gel entrapment formation by precipitation. Gels may be formed by precipitation of some natural and synthetic polymers by changing one or more parameters in the solution, such as temperature, salinity, pH of solvent, etc. Several materials can be used for entrapment. The examples include methods involving thermal treatment. Some disruption of viability can occur naturally. Next method is entrapment in preformed structures. Hollow fiber reactors can be used to immobilize plant cells by entrapment. The cells are placed on the shell side of the reactor and nutrient medium is rapidly recirculated through the fibers. This may have important applications in large scale. In other examples, the cells are added to preform polymerized structures such as polyurethane foam. When cells and suspension are mixed with these materials, they are rapidly incorporated into the network and subsequently grown into the cavities of the mesh and are entrapped by physical restriction and attachment to the matrix material. The mechanism of this involvement is at first a mechanical entrapment and later the fixation of the cells due to mechanisms of adsorption and adhesion or even due to their natural tendency for aggregation. These methods of immobilization have several advantages over other methods in that they are simple, cheap, gentle and rapid and maintains cellular functions. Next method is surface immobilization. Surface immobilization may occur on both natural and other matrices. Examples of natural matrices are deeper callus layers and cellulose, while synthetic one includes nets of steel and nylon. For example, cells of Solanum aviculaire were covalently linked to beds of polyphenylene oxide which had been achieved using glutaraldehyde by Archambault et al. In 1986. They described the spontaneous and rapid binding of Chamaeranthus roseus cells to a man-made material. Next method is immobilization by embedding. The temperature-dependent solubility of macromolecules like agarose, agar and carrageenan or the differing solubility of the sodium and calcium salts in the case of alginate are utilized to form polymeric gels or gel combination. Insoluble are formed under cold conditions such as agar or in aqueous calcium chloride solutions that is alginate. Their structure is non-uniform with differing pore diameters at the surface and in deeper layers. The size and form of the beds can be determined in part by stirring speed and using alginate, by the viscosity of the solution and dropping aperture. Next module is study of different enzymes. We will start with hyaluronidase. Hyaluronidases are a family of enzymes that catalyze the degradation of hyaluronic acid, that is HA. Carl Mayer classified these enzymes in 1971 into three distinct groups, a scheme based on the enzyme reaction products. The three main types of hyaluronidase are two classes of eukaryotic endoglycosidase hydrolases and a prokaryotic lyase type of glycosidase. In humans, there are 5 functional hyaluronidases. First is HYAL1, HYAL2, HYAL3, HYAL4 and last is HYAL5, also known as SPAM1 or PH20, plus a pseudogene HYAL6, also known as HYALP1. The genes for HYAL1 to 3 are clustered in chromosome 3, while HYAL4 to 6 are clustered in chromosome 7. HYAL1 and HYAL2 are the major hyaluronidases in most tissues. GPI anchored HYAL2 is responsible for cleaving high molecular weight HA, which is mostly bound to the CD4 receptor. The resulting HA fragments of variable size are then further hydrolyzed by HYAL1 after being internalized into endolysosomes. This generates HA oligosaccharides. According to their enzymatic mechanism, hyaluronidases are hyaluronoglycosides, that is, they cleave the 1 to 4 linkages between N-acetylglucosamine and glucuronate. The term hyaluronidase may also refer to hyaluronoglucuronidases, which cleave 1 to 3 linkages. In addition, bacterial hyaluronate lyses may also be referred to as hyaluronidases, although this is uncommon. Now its use as drug. By catalyzing the hydrolysis of hyaluronone, a constituent of the extracellular matrix, that is ECM, hyaluronidase lowers the viscosity of hyaluronone, thereby increasing tissue permeability. It is therefore used in medicine in conjunction with other drugs to speed their dispersion and delivery. Common applications are ophthalmic surgery in combination with local anesthetics. It also increases the absorption rate of parenteral fluids given by hypodermoclysis and is an adjunct in subcutaneous urography for improving resorption of radiopaque agents. Hyaluronidase is also used for extravasation of hyperosmolar solutions. Penicillinases. Beta-lactamases are enzymes produced by bacteria, also known as penicillinase, that provide multi-resistant to beta-lactam antibiotics such as penicillins, cefamycins and carbapenems, that is ertapenem. Although carbapenems are relatively resistant to beta-lactamases, beta-lactamase provides antibiotic resistance by breaking the antibiotic structure. These antibiotics all have a common element in their molecular structure, a 4-atom ring known as beta-lactam. Through hydrolysis, the lactamase enzyme breaks the beta-lactam ring open, deactivating the molecule's antibacterial properties. Beta-lactam antibiotics are typically used to treat a broad spectrum of gram-positive and gram-negative bacteria. Beta-lactamases produced by gram-negative organisms are usually secreted, especially when antibiotics are present in the environment. Penicillinase is a specific type of beta-lactamase showing specificity for penicillins, again by hydrolyzing the beta-lactam ring. Molecular weights of the various penicillinases tend to cluster near 50 kilodaltons. Penicillinase was the first beta-lactamase to be identified. It was first isolated by Abraham and Chain in 1940 from gram-negative E. coli, even before penicillin entered clinical use. But penicillinase production quickly spread to bacteria that previously did not produce it or produced it only rarely. Penicillinase resistant beta-lactams such as methicillin were developed, but there is now widespread resistance to even these. Now coming on to molecular classification of penicillinase. The molecular classification of beta-lactamases is based on the nucleotide and amino acid sequences in these enzymes. To date, 4 classes are recognized A to D, correlating with the functional classification. Classes A, C and D act by a serine based mechanism, whereas class B or metallo-beta-lactamases need zinc for their action. Penicillinase was discovered in 1940 and renamed beta-lactamase when the structure of the beta-lactam ring was finally elucidated. Detection. Beta-lactamase enzymatic activity can be detected using nitrocephin, a chromogenic cephalosporin substrate which changes color from yellow to red upon beta-lactamase mediated hydrolysis. Now coming on to our next enzyme, which is streptokinase. Streptokinase or SK is a medication and enzyme. As a medication, it is used to break down clots in some cases of myocardial infarction, that is heart attack. pulmonary embolism and arterial thromboembolism. The type of heart attack it is used in is an ST elevation myocardial infarction. It is used by injection into a vein. Side effects include nausea, bleeding, low blood pressure and allergic reactions. A second use in a person's lifetime is not recommended. While no harms have been found with use in pregnancy, it has not been well studied in this group. Streptokinase is in the antithrombotic family of medications and works by turning on the fibrinolytic system. Streptokinase was discovered in 1933 from beta hemolytic streptococci. It is on the World Health Organization's list of essential medicines, the most important medications needed in a basic health system. The wholesale cost is between $30 to $138 per dose as of 2014. It is no longer commercially available in the United States. Medical uses. If percutaneous coronary intervention, that is PCI, is not available within 90 to 120 minutes of first contact, streptokinase is recommended intravenously as soon as possible after the onset of a ST elevation myocardial infarction, that is STEMI. As streptokinase is a bacterial product, the body has the ability to build up an immunity to it. Therefore, it is recommended that this medication should not be used again after 4 days from the first administration, as it may not be as effective and can also cause an allergic reaction. For this reason, it is usually given only for a person's first heart attack. Further thrombotic events could be treated with tissue plasminogen activator, that is TPA. Overdose of streptokinase or TPA can be treated with aminocaproic acid. Next enzyme is streptodornase. Streptodornase, an enzyme produced by some hemolytic strains of Streptococcus that dissolves fibrinous secretions from infections, used medicinally, often in combination with streptokinase. A DNA hydrolyzing enzyme produced by hemolytic streptococci that is used medically, often in combination with streptokinase, to dissolve pus and fibrinous clots or deposits. Streptococcal deoxyribonuclease, that is streptodornase, has been characterized as a deoxyribonuclease of the endonuclease type, cleaving the 3' phosphate bond and producing 5' terminated fragments of variable length. Only traces of mononucleotides are produced together with small amounts of dinucleotides. The majority of the fragments are larger than dinucleotides. The total number of bonds cleaved is approximated as 18%. Next enzyme is amylase. An amylase is an enzyme that catalyzes the hydrolysis of starch into sugars. Amylase is present in the saliva of humans and some other mammals, where it begins the chemical process of digestion. Foods that contain large amounts of starch but little sugar, such as rice and potatoes, may acquire a slightly sweet taste as they are chewed because amylase degrades some of their starch into sugar. The pancreas and salivary gland also makes amylase, that is alpha amylase, to hydrolyze dietary starch into disaccharides and trisaccharides, which are converted by other enzymes to glucose to supply the body with energy. Plants and some bacteria also produce amylase. As diastase, amylase was the first enzyme to be discovered and isolated by Anselme Payen in 1833. Specific amylase proteins are designated by different Greek letters. All amylases are glycoside hydrolases and act on alpha-1,4 glycosidic bonds. Now coming on to their classification. First is alpha amylases. The alpha amylases, its alternative names are 1,4-alpha-D-glucanohydrolase or glycogenase, are calcium metalloenzymes completely unable to function in the absence of calcium. By acting at random locations along the starch chain, alpha amylase breaks down long chain carbohydrates, ultimately yielding maltotriose and maltose from amylose, or maltose, glucose and limit dextrin from amylopectin. Because it can act anywhere on the substrate, alpha amylase tend to be faster acting than beta amylase. In animals, it is a major digestive enzyme and its optimum pH is 6.7 to 7.0. In human physiology, both the salivary and pancreatic amylases are alpha amylases. The alpha amylases form is also found in plants, fungi such as Ascomycetes and Basidiomycetes, and bacteria such as Bacillus. Beta amylase. Another form of amylase, beta amylase, its alternative name is 1,4-alpha D-glucan maltohydrolase or glycogenase, saccharogen amylase, etc. is also synthesized by bacteria, fungi and plants. Working from the non-reducting end, beta amylase catalyzes the hydrolysis of the second alpha 1,4 glycosidic bond, cleaving off 2 glucose units, that is maltose, at a time. During the ripening of fruit, beta amylase breaks starch into maltose, resulting in the sweet flavor of ripe fruit. Both alpha amylase and beta amylase are present in seeds. Beta amylase is present in an inactive form prior to germination, whereas alpha amylase and proteases appear once germination has begun. Many microbes also produce amylase to degrade extracellular starches. Animal tissues do not contain beta amylase, although it may be present in microorganisms contained within the digestive tract. The optimum pH for beta amylase is 4.0 to 5.0. Now coming on to gamma amylase. Gamma amylase, its alternative name says glucan 1,4-alpha glucosidase, amyloglucosidase, exo 1,4-alpha glucosidase, glucoamylase, lysosomal alpha glucosidase, 1,4-alpha D-glucan glucohydrolase, etc. will cleave alpha 1,6 glycosidic linkages as well as the last alpha 1,4 glycosidic linkages at the non-reducing end of amylose and amylopectin, yielding glucose. The gamma amylase has most acidic optimum pH of all amylases because it is most active around pH 3. Now coming on to medical uses. Amylase also has medical applications in the use of pancreatic enzyme replacement therapy, that is PERT. It is one of the components in Solipura lipotamase to help in the breakdown of carbohydrates into simple sugars. Other uses. An inhibitor of alpha amylase called phaseolamin has been tested as a potential diet aid. When used as a food additive, amylase has E number E1100 and may be derived from swine pancreas or mold mushroom. Bacillary amylase is also used in clothing and dishwasher detergents to dissolve starches from fabrics and dishes. Factory workers who work with amylase for any of the above uses are at increased risk of occupational asthma. 5 to 9% of bakers have a positive skin test, and a fourth to a third of bakers with breathing problems are hypersensitive to amylase. Now coming on to next enzyme, which is proteases. Proteases refers to a group of enzymes whose catalytic function is to hydrolyze or break down proteins. They are also called proteolytic enzymes or systemic enzymes. Proteolytic enzymes are very important in digestion as they break down the peptide bonds in the protein foods to liberate the amino acids needed by the body. Additionally, proteolytic enzymes have been used for a long time in various forms of therapy. Their use in medicine is notable based on several clinical studies indicating their benefits in oncology, inflammatory conditions, blood rheology control and immune regulation. Read more about proteases in enzyme therapy. Protease is able to hydrolyze almost all proteins as long as they are not components of living cells. Normal living cells are protected against lysis by the inhibitor mechanism. Parasites, fungal forms and bacteria are protein. Viruses are cell parasites consisting of nucleic acids covered by a protein film. Enzymes can break down undigested protein, cellular debris and toxins in the blood, sparing the immune system this task. The immune system can then concentrate its full action on the bacterial or parasitic invasion. Now coming on to classification. Proteases can be classified into 7 broad groups. First is serine proteases using a serine alcohol. cysteine proteases using a cysteine thiol, threonine proteases using a threonine secondary alcohol, aspartic proteases using an aspartate carboxylic acid, glutamic proteases using a glutamate carboxylic acid, metalloproteases using a metal, usually zinc, asparagine peptide lyases using an asparagine to perform an elimination reaction not requiring water. How protease deficiency can affect your health? Acidity is created through the digestion of protein. Therefore, a protease deficiency results in an alkaline excess in the blood. This alkaline environment can cause anxiety and insomnia. In addition, since protein is required to carry protein-bound calcium in the blood, a protease deficiency lays the foundation for arthritis, osteoporosis, and other calcium deficient diseases. Because protein is converted to glucose upon demand, inadequate protein digestion leads to hypoglycemia, resulting in moodiness, mood swings, and irritability. Protease also has an ability to digest unwanted debris in the blood, including certain bacteria and viruses. Therefore, protease deficient people are immunocompromised, making them susceptible to bacterial, viral, and yeast infections, and a general decrease in immunity. Now coming on to our last module, which is conclusion. The technology of immobilized enzymes is still going through a phase of evolution and maturation. Evolution is reflected in the ever-broadening range of applications of immobilized enzymes. Maturation is mirrored in the development of the theory of how immobilized enzymes function and how the technique of immobilization is related to their primary structure through the formation and configuration of their three-dimensional structure. There still remains much room for the development of useful processes and materials based on this hard-won understanding. Although the science of enzyme immobilization has developed as a consequence of its technical utility, one should recognize that the advantages of having enzymes attached to surfaces have been exploited by living cells for as long as life has existed. An inquiry into the biological role of enzyme immobilization may provide some lessons for biotechnologists and serve as a second point of departure in addition to the purely chemical one. In fact, there is experimental evidence that the immobilized state might be the most common state for enzymes in their natural environment. The attachment of enzymes to the appropriate surface ensures that they stay at the site where their activity is required. The immobilization enhances the concentration at the proper location, and it may also protect the enzyme from being destroyed. Multimolecular assembly depends normally on weak non-covalent forces and hydrophobic interactions, but sometimes on covalent bonds as well. For example, disulfide bridges. All these different forces have been exploited in the development of immobilized enzymes. Immobilized enzymes will clearly be more widely used in the future. This is just the beginning of the immobilized enzyme technology era. With this, we have come to end of today's lecture. Hope you enjoyed it. This is Dr. Anshul Garg signing off. Till then, keep studying. Goodbye.